Nerve tissue cradle. On the 22nd, 23rd day of human embryogenesis, notochord's inductive factors cause the thickening of the median part of the ectoderm. The thickening is termed the nerve plate, which becomes a source of neural tissue. The lateral edges of the neural plate protrude above the surface of the ectoderm to form neural folds. With the neural groove between them. The neural folds converge, meet, and fuse along the median line, forming the neural tube, which is separated from the ectoderm. The cavity of the neural tube is preserved in adults as the ventricular system of the brain and the central canal of the spinal cord. Many neural fold cells remain outside the neural tube, forming the neural crest, located between the neural tube and the ectoderm. The process of the neural tube and the neural crest formation is called neurulation. Initially, the neural folds fuse in the lower cervical and upper thoracic region. Thus, the closure of the neural tube proceeds rostrally and caudally. The ends of the neural tube, the last to close, are called anterior and posterior neuropores. As the neuropores close, the neural tube undergoes transverse subdivisions, and the brain and spinal cord begin to develop. Initially, the brain exhibits three primary vesicles: the forebrain or prosencephalon, the midbrain or mesencephalon, and the hindbrain or rhombencephalon, with the secondary brain vesicles developing later. The prosencephalon splits into the telencephalon or cerebrum and the diencephalon. The mesencephalon remains as the midbrain, and the rhombencephalon develops into the metencephalon, the pons and the cerebellum, and the mielencephalon, or medulla oblongata. The neural tube consists of one layer of columnar neuroepithelial cells continuous from the luminal surface of the neural tube to the outside surface. They are rapidly dividing neural stem cells. Their nuclei lie at different heights, giving the impression that the neural tube has several cell layers. The nuclei move up and down within the cell cycle. Mitosis takes place when the nucleus is located at the part of the cell adjacent to the lumen, whereas S phase occurs while it is at the part adjacent to the outside surface. After mitosis, one daughter cell stays in the germinal layer, while the other one migrates away. The migrating cells form a second layer, which becomes progressively thicker as more cells are added to it. This layer is called the mantle or intermediate zone, and the germinal epithelium is now called the ventricular zone. The mantle zone cells differentiate into the neuroblasts and the glioblasts. The neuroblasts turn into neurons, and the latter send their processes, axons, away, creating the marginal zone. The mantle zone containing the neuronal cell bodies becomes the gray matter of the CNS, while the marginal zone turns into the white matter. The remaining neuroepithelial cells of the ventricular zone differentiate into ependymal cells, forming the lining of the ventricular system of the brain and the central canal of the spinal cord. The neural tube differentiates into neurons and macroglial cells of the CNS. The neural crest cells migrate to the periphery and develop into the craniofacial connective tissue, cartilages and bones, some parts of the heart and large arteries, and various cell types, melanocytes, odontoblasts, parafollicular cells of the thyroid gland, chromaffin cells of the adrenal medulla, and periganglia. In this chapter, it is important to note that cells of the neural crest differentiate into neurons and gliocytes in sensory ganglia of cranial nerves 5, 7, 9, and 10, dorsal root ganglia, autonomic ganglia, in Schwann cells in all peripheral nerves, and olfactory and sheathing cells. As it was noted, the mental zone cells of the neural tube differentiate into neuroblasts and macroglial cells of the CNS. Neurons develop from neuroblasts, which initially produce two short processes at opposite poles. One of these processes continues to grow to become an axon. The other one undergoes atrophy, being replaced later by dendritic tree. Thus, multipolar neuron is formed. The neuroblasts, migrating from the neural crest, differentiate not only into multipolar neurons of autonomic ganglia, but also into bipolar neurons of sensory ganglia. That is, only one dendrite appears instead of dendritic tree. In the cochlear and vestibular ganglia, neurons remain bipolar, and in other sensory ganglia, they become pseudo-unipolar. More detailed information on the development of neuroglia will be found in the following chapters. Thank you for the attention.